these are the signal transduction process, and because of that many reactions takes place, means numerous. If you will go back to all your last year pharmacology, whatsoever you have studied, so whatsoever adrenergic, cholinergic, etc., even if GPCR, you will say there are not less than 800 families of GPCR, and they are playing role, or 800 type of receptors are there. So similarly there are many receptor, many ion channels. So everywhere any change which has been brought in, even any kind of conformational changes they are bringing in, that can result into some alteration or some impact of it. Okay, it can be positive, it can be negative, or as we call it, it can be excitatory, it can be inhibitory, it can be stimulatory, it can be inhibitory, whatsoever we call it. And because of that there are some functions which are achieved, the function which are planned, they are achieved. So thus signal transduction, it plays very very important role in human physiology. So when these signal pathways are interacting with another, they form the networks. They allow the cellular responses to be coordinated, and they make the combinatorial signaling events. Again, for that, for your understanding, if you go back, particularly if you bring Rangan, Dale, or Goodman and Gilman, you will find that how even though we say that adrenergic or cholinergic, at many places they are opposite to each other, but you will find that they are coordinating, they are regulating each other's signal. So similarly these signals are there, they are regulating each other's signal, they are making a combination of it, and these they includes many things. So it can be transcription, it can be translation of genes, it can be replication, or it can initiate replication, because during replication also we have seen that some signal comes because of these signals. Example I told you yesterday, there are growth factors. Suppose these growth factors will come, growth factors will cause, and they will initiate one or other action, and because of this growth factor and related actions there will be impact on the new cell formation. So this is how the cells are formed, this is how the activities are there. So there are many activities are there, as I told you, transcriptional, translational, post-translational, conformational changes, as I told you in protein. Any change, example, when we say that voltage gated sodium channel ion opens, that means conformational changes. So there are so many activities that are taking place, and because of that, the impact of that, some other activities are there. Example, if we say that there is a conformational change, therefore sodium channel is opening, sodium channel causes opening of maybe suppose calcium channels, and it causes muscle contraction. So like that, one after another, this is a cascade of reaction, one after another something happens. So it can control basic mechanism maybe of tissue contraction, relaxation, cell growth, proliferation, metabolism, whatsoever you think about, everything is happening because of this. So signal transduction plays very very important role. It is important communication tool, and because of that various activities are there. Now you know how it happens. Some ligands, some biological moiety is there, which may be hormone, which may be neurotransmitter, which can be simple chemical, it can be cytokine, as I told you earlier, it can be prostaglandin, whatsoever it may be. So these chemicals are there. These chemicals, we call them as a ligand. They have ability to bind with certain receptors. So they will bind with the receptor, they are the first messenger. Then they may or may not, if it is just simple ion channel, then they may not. If it is just excitatory or inhibitory type of neurotransmitter, so they may not involve any kind of second messenger. But in other cases we have seen that it can be second messenger, it may be IP3-DAG pathway, it may be protein kinase or cyclic AMP pathway. There are second messenger, thereafter it may be calcium, if it comes then it can be third messenger. So there are many such messengers are there, and thus one after another the molecules are coming, and thus the signals they are amplified, amplified, or they can be reduced, or this is the modulation of signal, and this modulation of signal will be taking place, and one after another such activity will be there. Fortunately, as the science growing, we call it as a computational biology, then various analytical tools. Because of that, we are able to understand how this is happening, what is happening one after another, and what can be role it may be playing, and it is giving us many ideas. This is because of the ideas only we have got a pharmacology subject. Okay, we understood that yes, in some persons there can be overacting, over activity of renin-angiotensin. Because of that, whatsoever problems are there, they may having hypertension. So we are having the drugs which can take care of hypertension. So this is how the functions are there. We are understanding it, and we can modulate it. We can have the ligands which can act on that, which can MAD. What can be stimuli? Stimuli can be many things. As I told you, it can be anything. If you remember first year anatomy physiology, then whatsoever neuronal signals are there or humoral signals are there, it can be any neurohumoral signal. If you remember, it may be example baroreceptor, it may be chemoreceptor, which is sensing carbon dioxide. These are the simplest form. Okay, anything which can be there. In diuretics we have seen that there is a juxtaglomeralar apparatus which senses the sodium content. So at every places there are so many biosensors are present. One more topic is there after this is a biosensor. So we can have this discussion simultaneously, that there can be many biosensors are there which are doing doing the sensing work. And you just go through anatomy physiology, it will give you idea. If you have seen thyroid, the positive and or all hormones, then negative control, positive feedback mechanism, everything is there. Without sensor it will not work. So there are many sensors are there. According to that, there are some time bound manner sensors there, even though if it is not there. Just like some machines are having timer are set, like the timers are set. Example, puberty is there, so it has got some timer set, and on that basis the puberty is happening at certain time span. Menopause is happening in females at certain time span. So these are all timer set actions. So these stimuli are inbuilt stimuli which are not seen. So there are many such things are there. It can be present extracellularly, it can be present intracellularly. Extracellularly it can be growth factors, it can be cytokines, many things we have seen earlier. So this can be various signals, or it can be DNAs which are giving signal, and all these are giving. We know ligands. I will not discuss about ligand, what are various kind of receptors. You know about these ligands. We know ligands like ligands which can bind adrenergic receptor or GPCRs or cholinergic receptors. So all are there. We know various kind of receptors. I will not go into this. This receptor may be on surface receptor, or it may be intracellular receptor. There are many extracellular receptors for almost all this, and these are drugs. They are for growth factors, cytokines, neurotransmitters, etc. There can be many receptors which are inside, and these are for various hormones, example steroid hormones and various nuclear hormones, and these are all we have seen them. You have studied it. Okay, if not, please go through again. And all these receptors, how they are working, you know about the mechanism. They are a very important part of signal transduction mechanism. So this is definitely part of your syllabus, but I am not teaching it because it has already been taught to you earlier. What I'll be teaching you is whatsoever which has not been taught to you, because we don't have much time for that. Already we have taken some extra classes to beyond what we have planned for this, for sale and all. So you all go through this, all these GPCRs, then membrane bound receptors, then nuclear receptors. You go through the mechanism, and some additional receptor which we shall be seeing here only it will be there. So there are many classifications are there. There are many ways there. So we have got multiple way of classification on the basis of size or chemical structures like neuropeptides, etc., on the basis of their actions, adrenergic, etc., where they are working. So there are variety of such receptors, and these are very important receptors, and on that various ligands are binding. The ligands are playing an important role in signaling, and thus this signaling play important role. There are certain receptor also there which are there in particularly breast cancer, HER, that is human estrogen receptor 2. They are capable of ligand independent activation or overexpress or mutated, and they are implicated in many of the pathological conditions. Okay. Then what are the mechanotransmissions? I told you touch, etc., and there are many such way by which it is there. Then there are, apart from that, the adhesion molecules are there in immunology. We have not studied that depth, but there are adhesion molecules are there, and there are many adhesion molecules are working. In that you will study all these integrins and which are acting on various cytoskeleton. So they will do it, and this cell adhesion molecule are example of these are cathedrins, lectins. They are also mediate mechanotransduction, and other forms of are there within nervous system are for hearing, touch, proprioception and balance, which you might have studied earlier. Okay, again during first year anatomy physiology, all these are mechanosensation. Hearing is a mechanosensation. Why? Because waves are the sound waves are having impact on tympanic membrane, and because of the vibration is there. Touch is also mechanosensation. Pressure, proprioception, that where the body parts are located, they are there. Balance is a mechanoception. So these are various mechanical sensation we have. Then osmotic pressure, you have recently studied in diuretics that how osmotic pressure is sensed, and on that basis how it is there. Or similarly chemoreceptors are working. Temperature are also thermoreceptor, and there are variety of molecules are there. Very important are, they are called as some are known as a heat shock proteins, HSPs. Heat shock protein 40, 70, 90s are there, and these proteins are found and they become active, and they are responsible for some of the target genes. They playing important role in various pathological conditions. Okay. Then light, already visual, I told you, rhodopsin cycle and all. So these are the receptors. We have seen about it, we know about them. Example of these are extracellular receptors. We know about, there are variety of extracellular receptor. Maybe these are membrane bound channel receptor, maybe they are the G protein coupled receptor, maybe they are tyrosine kinase type of receptors. There are variety of receptors are there. You know about it, and I will not go into this detail how these receptors are working. You can have it from your earlier studies. So these all receptors are there. I will not explain this. As I told you, there are more than 800 types up there, and if see 800 members are there in human beings. In other animals, if we count, then it becomes more than 5,000 type. And there are many major 5 families are there in GPCR: rhodopsin like, secretin like, metabotropic glutamate receptor, adhesion molecules and Fiesel type. These are basically 5 families are there, and in that this whole 800s are divided. Those who are interested can see a very good tree has been put into Goodman and Gilman. So they can see this around 830 or 840 receptors, how they are. Again, all the details already been studied by you people, that how these GPCRs are there, how these GPCRs are working, what kind of subunit alpha, beta, gamma are there, what kind of G proteins are there, G0, GI, GQ, etc., and what are the further consequences of that. Then next are known as the tyrosine kinases, or they are also called as a receptor tyrosine kinases. RTKs, they are transmembrane proteins. Earlier they were known as insulin like growth factor receptor. IGRF was a common name, later it has been changed. It has been found that these are all the protein kinases. Insulin also having similar type of receptor. These are the dimers, and when the ligand binds, then these dimers come together, both subunit come together, and then they will causes the further cascade of reaction. And many internal proteins, just like in G protein you have seen IP3DAJ or cyclic AMP, whatsoever you have seen. Similarly here various kind of phosphorylation are there, activation of protein will be there. Example of these are known as a serine threonine kinases, They are important in signal transduction, particularly in apoptosis and other pathways. Then these are also having important interaction with the G proteins, like these are known as RAS kinases, Rho kinases, and RAF families of receptors, and they are collectively called as a small G proteins. So they are having, some what we can say similarity between G protein also and protein kinases also. At the molecular switches, usually they are having this isoprenyl group is linked to their carboxyl end, and these assign proteins to the specific membrane subdomains. So this is how they are acting. Why we are having one what we can say addition over here. So these are important. This mutation of some of these RTK genes or some of the GPCRs, they can result into the expression of receptor which are existing constitutively in activated state, and these mutated genes, they may act as oncogenes. Oncogenes means the genes which are responsible to cause cancer. So this therefore this signal transduction, any part, these are all part of the transaction. So ligand is the one part, the receptor is there, and all the transduction factors are there, or whatsoever its components of the receptor activation mechanism is there. Till last part, everything is the part of this cascade mechanism. So there are some histidine specific protein kinases are also there, which are structurally distinct from other protein kinases. They are found in various prokaryotes, fungi and plants, which is part of two component signal transduction mechanism, and this is important in their internal cell signaling in the prokaryotes. Then important variety of this kind of receptors is integrin receptors. They are extracellular type of receptors. They are connected with fibrinolactin and collagen, and ligand binding to the extracellular domain integrin changes the protein conformation. The cell membrane initiates signal transduction. They lack kinase activity. The mediated signal transduction is achieved through variety of intracellular protein kinases and adapter molecules. We call them as integrin link kinases, okay, ILKs. So these internal link kinases are there. They are causing activation of various things, and thus they cause cooperative integrin and this receptor tyrosine kinase signaling. It will determine the timing of the cellular survival, apoptosis, proliferation and differentiation. I hope you are understanding the importance of this, that this RTK signaling, this integrin RTK signaling is determining the timing of cellular survival. So how long the cell should survive, or when the cell death should occur, or when the apoptosis should occur, because we have seen apoptosis is the process which causes the destruction of the damage, or destruction of the damaged cell, the cell itself, or which are damaged. Then by apoptosis they are totally removed, they are killed. Okay. Then proliferation means growth, and differentiation, that means going into a different function. So like that, this particular function is done through the integrins. So when the ligands, they are binding with the integrins, they will be having action. See, you can see this RTK is a dimer. So when this ligand binds, see this and this both are combined together, otherwise they were separated. So they come together, and then this is the pathway. This is called as a signaling pathway. So once this is activated, because of that RAS is activated, RAF is activated, it causes activation of MEK, it is activation of MAPK, that is mitogen activated protein kinases. Then these are the AKT is a protein kinase B, and these are various kind of that is phosphatidylate inositol triphosphate IL, that is we have seen integrin link kinases proteins. So these all are there. So many proteins are there, FAK, SRC, CAS, CRK, RAC. These all are involved in either survival, apoptosis, proliferation, or differentiation in the nucleus. So this all signaling gives information to the nucleus, and accordingly the nucleus, that means the genes and the chromosome, they will decide what to do, whether should go for proliferation, that means replication, etc., or go for differentiation, that means a specific activity which was not earlier been shown should be brought into, should go for survival, or apoptosis means death of the cell. So these are the important signal transduction mechanisms which are important, and they are resulting into variety of activities. So this is one kind of signaling, that is integrin link kinases are there. Then next is toll-like receptor are also known. These are the adapter molecules within the cytoplasm of the cell, and they propagate the signal. Toll, toll gate are there, okay. So these adapters, they activate other intracellular molecules. They are like IRAK1, IRAK4, TBK, IKI, and they will amplify the signal, and they will lead to the induction or suppression of the genes. So that means what again I am trying to emphasize, that there are many mechanisms, different mechanism by which either genes are modulated, genes are either their expression is either increased or decreased. So these are the various ways by which they are doing. Ligand gated ion channel, you know about them, how the action potentials are generated, or how it can be EPSP or IPSP. IPSP means inhibitory postsynaptic potentials or excitatory, and you have seen them in muscle contraction, you have seen in cardiac contraction, you have seen in cardiac cardiac impulse conduction. At many places these ligand gated channels are there, and they are also playing important role in reaching to threshold potential in whether it is mechanotropic receptors or all. So this is all intracellular receptors. Again, they are known to you. These are having many steroid or lipid type of these hormones which are reaching into the inside the nucleus, and after reaching inside the nucleus, they will have many activity. This you have seen glucocorticoid mechanism at many places. Last year you have seen, or earlier you have seen in bronchial asthma. This year you have seen as a immunosuppressant. So at many places you have seen, you know about it, and if not, then you should have revised it. So these are the receptors. They will attach to DNA at a specific hormone receptor element, that is HRE sequences, and they will be located in the promoter gene, and accordingly either they will cause the gene expression. Gene expression word is used for any kind of genetic processing which is either for transcription or replication. So accordingly gene expression will be there, and this regulation of gene expression will have two consequences. So this is making the activities for constant this what we can say, either increase or decrease in the activities of particular type of gene, and accordingly the gene will be expressed. They also have DNA binding domains. They are containing zinc fingers. Already we discussed earlier, zinc fingers, they are small protein structure motif which is characterized by the coordination of one or more zinc ions, and that stabilize that particular folds of the zincs, and they are playing very important role in this gene expression. They stabilize DNA binding by holding its phosphate backbone, and DNA sequence that match the receptor are usually hexameric repeats. Okay, hexameric means 6 molecules are there, and the ligand binding domain is additionally responsible for dimerization of nucleic receptors prior to binding and providing structure for transactivation for the communication with the translation apparatus. So these are the subclass of the nuclear receptors, and there are many molecules. I told you about heat shock proteins are there. They can able to modify the gene activity, they can alter it, and then retinoic acid receptors are there. There are also another subset of receptors. There are a variety of such receptors are there which are playing important role. You know about second messenger. There are multiple second receptors. There is cyclic AMP, there is IP3, there is a DAJ, there is a calcium. So I will not discuss into it. At many places they are there, and how they are working you know. So these all second messengers are there which are playing important role, and they they are transmitting whatsoever ligand has transmitted to the receptor. From receptor these molecules will go for the next, and accordingly there will be either activation or deactivation of one or other activity, and therefore accordingly they will act as a excitatory or inhibitory. So they play important role. So there can be lipid messengers, there can be nitric oxide is a second messenger, because it is a free radical and can diffuse through the plasma membrane and causes the activation of soluble guanyl cyclase. That mechanism you might have seen last year. You have studied it in angina pectoris, organic nitrates and other drugs, how they act. So these are how important they are. So there are variety of activities we are bringing out. We call them as a cellular response because of the signal transduction, and that signal transduction, as I told you, or you know about it, there are multiple activities they are bringing out. You just imagine and you know that these all are activities they are happening because of one or other kind of cellular transduction. Okay, so you can make a big list about it, what kind of activities are there. There are many such receptors, and many activities are there which are at variety of places they are acting, and these all combination of all these signals, they are integrated in altered cytoplasmic machinery which leads to altered cell behavior. So this is what importance of the cell signaling, that the signals are integrated in an altered cytoplasmic machinery which leads to altered cell behavior. So what are the major pathways? Now these are all listed in your syllabus, that we shall have variety of this signaling pathways. I'll mention them, and then we'll have later one. So these are some of the important pathways. These are known as a MAPK or ERK pathway. MAPK, that is mitogen activated protein kinases. Okay, or this I am missing now, receptor kinases pathway. So it couples the intracellular response to the binding of growth factors to the cell surfaces receptors, and this pathway we shall be having slight more details. It promotes cell division, and many forms of cancer are associated with this type of receptor. Cyclic AMP dependent pathway, you have seen it at many places. You have seen last year all G protein couple, most of the action you might have seen it, and they are almost present in many cells. Cyclic AMP dependent pathway. IP3 DAG pathway also last year you have seen particularly in various adrenergic and cholinergic neurons, which is common one. These are very common one. Apart from that, there are many which are there, which we can see how they are working, how they will be playing role, and what kind of activities will be there. In immunology, they are playing very important role, because even during studying immunology, if you remember, we have seen about it, that whenever there is any kind of immune reaction is taking place, whether it is inflammation or because of the injury to the host cell as such, or because of the microorganism or any foreign object whenever it is coming, so that is result to antigen antibody complex, and then it causes it has been engulfed, causes initiation of action, or otherwise even there is any kind of injury, because of that various kind of autocoids, example prostaglandins, then various interleukins, leukotrienes and various chemokines, etc. They all are released, and they will cause activity, and there are many this one I told earlier, and I'll repeat it. I remember one book of immunology which says that immunology is the invention of devil. So there are so many type of receptors are there, so many mediators are there, and they are playing variety of role into it. Fortunately, whatsoever immunosuppressant drugs are there, we are almost they are linked with the interleukin 2 only, if you remember what we have studied. But there are many other receptors which are playing important role, and because of that they are showing variety of actions. Uh, in one or other two classes we shall try to have glimpses on variety of these receptors, how these receptors or how these are working, and then we shall go for the next topic whatsoever you finalize. But before that, what we will do is this is by this weekend we will be finishing this. I want now questions from you. I want to know that whether you are understanding, because only around 13-14 students are remaining present, and then it's not good actually that for these important topics you are not or students are remaining absent. So if you can make the questions, at least those who are attending, let me understand whether you are understanding it properly or not. So if it is so, then fine. Because whatsoever depth I have taught, I want to know that whether you are reading up to that depth and whether you can understand it or whether you are able to make the questions out of it. So I am expecting questions one after another topic wise, but you go reverse way now because you complete it on this cell related first and then go for other diuretics, immunology, etc. Okay, toxicology. I think this is fifth topic we are finishing. So go through this. As I told you, 1 or 2 lecture maximum we shall take and we'll finish this. These are very important one. If we understand, I expect if you can read it in detail, it will definitely help you a lot. And once you read it, that will definitely make a good impact on it. Okay, so all the best, make the full use of it.